Alright guys, today we'll be going over an EABS 2 review, an EABS 1 review, second session. Share my screen. Here we go. So the first chapter is about membrane dynamics. So what's most important about this section is that there's a little bit of a review, but for this one, it goes over the different components of ions within intracellular, extracellular, and interstitial space of a cell. So when you look at it and evaluate it, you'll notice that an intracellular fluid actually has more potassium. Interstitial fluid has more sodium, and as well as the plasma. And so those concentrations will be important later on when we discuss neurons and electrical activity that occurs within cells. Osmosis and tonicity, they're two different concepts, but some are related with each other. So the basic foundations for osmosis is that osmosis is the diffusion of water through passive means. And since we're talking about water specifically, we need to evaluate why the water moves. So water is attracted to solutes. So solutes in the body attract the water, and because of this water present there, you're going to have a buildup of pressure. And osmotic pressure is the equivalent pressure that you would need to counter the buildup. Here's an example of it. If you place, say, more solids on this side of a solution, then water will tend to want to balance out on both sides, have the same concentration of water. So for you to counter that osmotic pressure, you need to put an equivalent force downward. So osmolarity determines the number of particles in a solution. It talks about pretty much the concentration of particles, of solutes within a solution. And tonicity describes the volume change of a cell. So in terms of definition, tonicity describes a physiological change as to what would happen to the cell if you place it within a solution. And here we talk about hyperosmotic, hypoosmotic, and isoosmotic, as well as isotonic, hypotonic, and hypertonic. Not everything can penetrate through the cell, so the cell has penetrating and non-penetrating molecules. And this chart relates to conditions where actually tonicity and osmolarity could and could not be related. So if you're hypoosmotic, then chances are you could be hypotonic. If you're hypoosmotic, you're for sure hypotonic. But if you're, let's say, hyperosmotic, then you could be hypotonic, isotonic, and hypertonic. And so those are different conditions that you should know where regarding the osmolarity and tonicity. These are some general rules that you can follow when you get questions where you have to determine the hypertonicity or osmolarity in a solution, as well as some common solutions that are used today in the medical field, such as normal saline. So then we go over the transport processes. The transport processes involves, as a classification, passive and active transport. And so for passive transport, we have diffusion, through simple diffusion, and facilitated diffusion. And the difference between these two is that facilitated diffusion occurs through a mediation of a protein available in the plasma membrane, versus simple diffusion, where it just goes from regions of high concentration to low concentration. And these don't require energy, it just requires a gradient. And a gradient is just an unequal area of concentration. So we go from high to low. On the contrast, active transport requires ATP. And so that can occur through primary transport, as well as through other measures such as endocytosis, exocytosis, or phagocytosis that requires the plasma membrane to move its cytoskeleton in order to either engulf a bacterium or to create vesicles, which are pockets of the plasma membrane to carry proteins or other stuff. Talks a bit about the diffusion. And so when you look at the components that are able to diffuse, most of them are actually lipids. And that's because the plasma membrane is a lipid. It's a phospholipid bilayer. So nonpolar lipid molecules are preferable, and also in smaller sizes. As you increase in size, it becomes difficult to transport a molecule as a general rule. And protein-mediated transport occurs for molecules that are polar but are small. The same rules applies. As you get bigger in size, it becomes harder to transport a molecule through the plasma membrane. but small polar molecules are able to transport through the help of protein-mediated responses. And so these proteins help the cell in 4 general ways. It increases the structure, it serves as an enzymes, transporters, and receptors on the plasma membrane for certain chemical reactions. And they can be of two types, open and gated channels. So for gated channels, you can be either mechanically, or voltage gated, as you will learn later with neurons. Here's a chart that depicts the different types of channel proteins and carrier proteins. Channel proteins, open and close, and carrier proteins transport the molecules, but they can be unipore carriers, where it only carries one type, sympore carriers, where it transports different types, but only one way, or antipore carriers that transport different types, but in opposite directions. We look at active transport, and we understand that since it goes against the concentration gradient, it requires energy. Naturally, you will go from areas of high concentration to low concentration, but active transport does the opposite. It goes from regions of low concentration to high concentration. So this is what we call going against the gradient. And because you're going against the gradient, you need to input energy. So in this case, we input ATP. Some examples of active transports include ATPases. So these hydrolyze ATP into ADP in transport. They also include sodium-potassium pumps, sodium glucose transporters, and glucose transporters. So when a component might be too big to do either of those things, there are other ways to transport the component through vesicular transport. So vesicles are like pockets of the cell membrane that either indent inward or indent outward and create an extension of the plasma membrane to allow these components into the cell or out of the cell. Phagocytosis talks about when a cell indents outward to consume something. So it extends its plasma membrane outward and it engulfs it into this vesicle. And it usually does this for components that are foreign to it, such as other cells and bacteria. Endocytosis creates smaller vesicles. So compared to phagocytosis, the vesicles that endocytosis creates are smaller and they indent inward rather than pushing outward, but they achieve the same function in that it wants to transport something into the cell. And usually they're mediated through receptors, such as clathrin-coated receptors or caveolae, which are small cave-like structures. Similarly, if you want to transport something out of the cell, exocytosis will be a great option. So it's intended to be a continual process. Most cells that actually do exocytosis do so continually, and they don't stop, like goblet cells when they secrete mucus in the intestines. For chapter 6, we go over communication, integration, and homeostasis. And cells' communication can be either from a short or local distance or a long-term distance. And they can be of several types. Most known are chemical, electrical, and contact dependent or mechanical. So the most simple one is when two cells directly communicate with each other in cell-to-cell communication. And these communications are formed through gap junctions. So gap junctions allow for fibers called connexins to connect from one cell to the other and attach themselves. And this will create a protein channel called a connexon. And so this connexon is literally connecting these two cells into one cell. Other communications is contact dependent. If cells cannot create a connexon or don't have these protein fibers, then they need to physically touch in order to create communication. And this is usually done during embryonic stages of development, where cell adhesion molecules serve to carry out these local communications. Paracrine and autocrine signals are chemical signals. So every time you hear paracrine and autocrine, it just means that there's a chemical involved. And they can carry out local communication. And they travel through the bloodstream circulation. And so what these chemicals do is that they act on the receptors of the target cell. If you're going longer distances, then you might want to consider electrical and chemical communications. These as well also travel through the bloodstream or through the nervous system, and particularly involve other components like the neuromodulators that influence the effect of it, neurocrines, which are hormones that travel in the bloodstream, and cytokines. Cytokines are important because they act as both short and long-term signals, such as histamine and erythropoietin. Here's a diagram that explains the gap junctions that form in direct cell-to-cell contact, the contact-dependent signals, and autocrine signals that would spread over either short or long distances from a chemical to a receptor in a cell, and how an endocrine and nervous system's response would be initiated. When we look at signal pathways, you look at receptor proteins that are located inside the cell membrane. So for a response to happen, you need a receptor. And so that receptor receives the signal, the chemical messenger, and it can be received either on the plasma membrane surface or within the cell. And there are usually 4 categories of plasma membrane receptors: receptor channels, receptor enzymes, G-protein coupled receptors, and integrin receptors. And the one we'll go into detail today is the G-protein coupled receptor. And so this would allow the cells to communicate by receiving the signal and then processing it, sending out a secondary messenger. So first we have the receptor on the plasma membrane, and then after that we have a secondary messenger within the cell, and that sends a signal to the integrating center. Important thing to note, receptor enzymes have protein kinase or guanylyl cyclase activity. And so basically means that these receptors are usually divided into 2 components. One that is the receptor on the side of the extracellular plasma membrane, and within the intracellular is going to be an enzyme that acts on this response and sends a secondary messenger. Most signal transductions use G proteins, and so G proteins are G-protein coupled receptors that contain a G protein. They're special and unique because they wrap around, they penetrate through the plasma membrane 7 times and are connected to this G protein at the tail of it. And the G protein is a 3-part membrane, as well as these pathways. The most common pathways used to use these signal transductions to convert this chemical signal into a cellular signal is a GPCR CAMP pathways, which is a G-protein coupled receptors cyclic AMP pathways. Here's an example of the different components involved within signal transduction. So we first have the signal molecule, which is a chemical. It binds either to a channel or a membrane receptor. And from there on, it initiates a series of responses that begins with a first messenger and then goes on to a second messenger. The second messenger then targets the intended protein, and the intended protein initiates a cellular response, which would be one of these 4. Other molecules that influence signaling is calcium. Calcium is important in the nervous system, but as well as signal transduction. And when you have the presence of calcium within signal transduction, you usually would get one of these responses. When it's bound to calmodulin, it alters enzyme activity or transporter activity. It also is involved in the contraction of the cytoskeletal proteins like microtubules. And it also is involved in exocytosis of vesicles. It changes ion channels to change a gating state and initiates development in the embryo. Similar idea, you would have this case a chemical messenger, calcium, which would bind to voltage gated channels, and then initiates a secondary messenger that binds to proteins that initiates eventually a response. Gases as well can be signaling molecules, the most common of them being nitric oxide, carbon monoxide, and hydrogen sulfide. And these are generally used to relax and dilate blood vessels. Lipids as well can be important chemicals messengers, and the most important ones are the eicosanoids, which are derivatives of arachidonic acid. They are classified into 2 major groups, the leukotrienes and prostanoids. And how these pathways can be modulated? Well, one ligand can have multiple receptors, So a cell can have for sure multiple receptors, and they could be of different varieties of the same family. We call those isoforms. So you can have two different receptors, and they could be very similar, but have opposite effects. This is because of their isoform. Because the receptors compete for space within the cell membrane or within the cell, then there is competition for space, the reaction at which it binds to, the chemical messenger, so it's specific, and there's a maximum cap at which these receptors can occupy. And they're regulated to be sure they're kept in place by either downregulation or upregulation. So if you want downregulation, then you want to decrease the active receptors. And you can do this by either by doing endocytosis to physically remove them, or to deactivate it. So you will bind a chemical messenger that would bind to this receptor and then deactivate it, which is called an antagonist or desensitization. On the other hand, if you want to increase active receptors, then you produce more receptors. So you would produce the gene that, you would activate the gene that produces more receptors of this type, or you bind receptor agonists to it. So you would bind a chemical messenger that would reactivate it. Antagonist means disactivate, and agonist means activate in this context. When you consider the body, the different parts of the body, how they chemically or electrically communicate, you could see that there are different steps, intermediate steps, to carry out this message, and that it might span from the most simplest of pathways to the most complex of pathways involving several steps. Chapter 7, Introduction to the Endocrine System. So the endocrine system is important because it regulates chemical messaging. And so we need to know what a chemical hormone is. A chemical hormone is secreted by a group of cells. As we went over, they tend to travel in the bloodstream. Pheromones are a subclass of hormones that trigger response from other organisms. And what's unique about hormones compared to other things is that they exert their effects at low concentrations. So hormones need to bind a receptor to have an effect. And because of this, the receptors themselves could be situated at different regions of the body and have different effects depending on where it is. And hormone actions must end. When a hormone is circulated through the bloodstream, it usually ends up being deactivated by the liver and the kidneys, which will convert it into non-inactive metabolites. And so the time that it is spent in the body and how much time it will take for half of it, of the original amount, to be taken out of circulation in the body is called the biological half-life. And so it just gives us an idea of how, when we take, let's say, a new drug, how would it affect the user? Hormones could be of different types. The most common being peptide hormones is the most abundant in the body. Then there are also steroid hormones and amine hormones, which come from one amino acid. Peptide hormones are produced in the cell, and because of that, you first begin production in the ribosome, which produces proteins. Then you go to the Golgi apparatus. And so when it is produced in the ribosome, it is in an inactive state, this hormone. And later on, after it's packaged and produced by the Golgi apparatus, once this Golgi apparatus produces a vesicle around it and packages it, it converts into an active hormone and peptide fragments. And this is useful because it would allow the hormone to have a delayed effect and only activate when it is necessary, or when it travels to its target. When we consider lipid hormones, most of them tend to come from cholesterol. So most of them are actually cholesterol derivatives, these steroid hormones. And they are made in the adrenal cortex, ovaries, placenta, and testes. Because it is lipid, it is able to diffuse through the phospholipid bilayer. But when it travels in the bloodstream, it usually needs a protein carrier molecule to be able to transport it, since we're talking about a lipid within the bloodstream, which is polar. Some of the most common steroid hormones involved include corticosterone, aldosterone, testosterone, androstenedione, progesterone, estrone, but these all come from cholesterol. So when you compare their chemical structure, you would see that they're actually very similar. And this is the process by which it occurs. The steroid travels to the bloodstream, with a carrier protein, then it goes through the cell membrane or activates with a cell receptor on the cell membrane and initiates a response from the cell. So similar to the signal transduction, when you have the control of hormones, then you would have neurohormones. Oh yeah, the amino acid hormones. So amino acid hormones and peptide hormones, the only difference is that amino acid hormones is produced by one amino acid. They tend to be either tryptophan or tyrosine derived. Peptide hormones are produced by a chain of amino acids. And so when we're looking at amino acid hormones, we're mostly looking at melatonin, epinephrine, and norepinephrine. When we look at neurohormones, which are hormones involved with the nervous system, then we're talking about catecholamines, which are epinephrine, norepinephrine, those secreted by the posterior pituitary, and those secreted by the anterior pituitary. And the pituitary gland by itself is actually two fused glands. So the anterior pituitary is a true endocrine gland, and the posterior pituitary is an extension of the neural tissue of the brain. And we'll see that here in this diagram. We can see the anterior pituitary and the posterior pituitary. So the anterior pituitary produces 6 hormones. We have prolactin, growth hormone, thyroid stimulating hormone, adenocorticotropin, luteinizing hormone, and follicle stimulating hormone. And so one way I like to remember is this is flat PG, FLAT PG, as an acronym for all 6. When we look at the posterior pituitary, then we have oxytocin and vasopressin. And so the purpose for these hormones, the general purpose involves oxytocin, contraction of uterine muscles, vasopressin, the reabsorption of water and water homeostasis in the kidneys and in the blood. For prolactin, it controls milk production and immune system. For thyroid stimulating hormone, it controls synthesis and secretion of thyroid hormone in the thyroid glands. For adenocorticotropin, it regulates the hormones of cortisol in the adrenal cortex. Growth hormones, as the name implies, affects the growth production. So hormones that affect growth, follicle stimulating hormone and luteinizing hormone are classified as what we call gonadotropins. And so they mostly affect either the sex cells or the gonads. And so when we evaluate the anterior pituitary, we notice that it has a portal system. And so this portal system is basically a series of 2 capillary beds connected from the hypothalamus. And the benefit of having hormones that travel through this portal system is that it will carry a different effect because it will remain concentrated as it travels to its target. Feedback loops involved with these hormones are long loop feedback, long loop negative feedback, where the product of the whole pathway shuts off the secretion of a hormone. So it's funny because the end product stops the production of this pathway, or short loop negative feedbacks. So hormone interactions, when you have hormones, usually their effect combined are much bigger than when you add them separately. So we call this synergy. So because of the synergy between hormones, we would have the best effect possible. If one is lacking, then you would have a decreased effect. So one example would be if you have thyroid hormone and reproductive hormones together versus individually. If you have them individually, they might delay reproductive progress or reproductive growth. But individually would allow an organism to have normal reproductive growth, development. So antagonist hormones could be isomers of each other. Basically means that these isomers, which are similar structure, can have different effects. So one of them could be vasopressin alpha and beta. And what could go wrong when you have excess hormones? When we have too much hormones, it's called hypersecretion, and too little of it would be called hyposecretion. If the issue is not with the amount of hormone itself, then it's probably with the receptor that it receives this chemical messenger. It could be too many. It could be in a case where there's too many receptors, so the body would downregulate it, by binding antagonists to it to deactivate receptors. Or if there's too much receptors, then the body, or there's too little receptors, in the body could produce more and upregulate it. For chapter 8, we go over neurons and the cellular networks. So the nervous system is basically comprised of the central nervous system and the peripheral nervous system. And within the peripheral nervous system, we have a subsection, which is a sensory division and an efferent division. And the organs involved within these systems is the central nervous system will be the brain and the spinal cord, and the peripheral nervous system will be all other nerves in the body. If we evaluate the structure of neurons themselves, they're comprised of a nucleus. So this is what we call the cell body. So the cell body will be the integrating center that processes information. That processes information and initiates a response. Then there's an axon hillock. Once the cell decides that it's going to process information and propagate it, it propagates through the axon hillock down through the axon, which is myelinated. This myelination allows for the neuron to expedite its propagation and reaches towards the end, which is called the axon terminal. At the axon terminal, there's a small gap, which we call synaptic cleft. These axons are really not connected to each other. There's always a small gap, and this small gap allows for the exchange of what we call neurotransmitters, which are the chemicals that exchange the information. And so when we look at these neurons can typically be, they typically have these landmarks, the axon, the cell body, and the dendrites, and they can be multipolar. So this regards to the shape, they can be multipolar, which is the most common one. This picture is a multipolar neuron. They could be pseudo-unipolar, or they could be bipolar, in which they have a single axon and dendrites. And they could be interneurons, which are found in the spinal cord, sensory neurons, or motor neurons. So sensory neurons bring information about what you feel through your senses. It is integrated in the spinal cord or in the brain, and then it is sent a response through motor neurons. So one unrecognized or underestimated group of cells that provide a lot of support in the nervous system are the glial cells. So glial cells actually outnumber neurons 10 to 50. So then within glial cells, we have subsections, and they're thought to provide different aid either by building myelin sheaths or by removing chemical substances when there's action propagation. We have Schwann cells and oligodendrocytes. They're the same thing. The only thing that changes is their region in which they're involved. When you have Schwann cells, you're talking about the peripheral nervous system. And when you have oligodendrocytes, you're talking about the central nervous system. And so these protect the neurons by forming this fatty lipid myelin sheath around it that serves as insulation as well as action propagation. Satellite cells, they're non-myelinated Schwann cells, but what they do is that they form a knot around the cell body. So it's not a myelin sheath, but it still provides some protection. Astrocytes, they transport chemicals in the synaptic cleft. And microglia, they remove damaged cells and other foreign invaders from the body. This could have also a negative effect in the sense that when microglia are under stress, they release oxidative free radicals. So it's thought to believe that this phenomenon occurs whenever people are actually under severe stress. The release of the free radicals, which hurts local neuron growth. And you have ependymal cells. Ependymal cells are important because it creates a layer that protects the fluid compartments within the central nervous system. Here's a picture with an overview of the different types. You have ependymal, astrocytes, microglia, oligodendrocytes, Schwann cells, and satellite cells. Then we talk about the electrical signals within the neuron. So neurons are actually composed of different ions, and the way the electrical signal propagates is through these ions. So these ions carry a charge, but they by themselves are also chemicals. So when we evaluate the potential present for a single ion species, we would use a Nernst equation. So in this case, let's say it would be for sodium. We can use the Nernst equation for sodium and determine the electrical potential present for sodium. But since we know that nerves and neurons actually involve several species of ions like potassium, sodium, chlorine, then we remodify this Nernst equation to include those species, and we get the Goldman-Katz equation. And these are the conditions for it. Neurons by themselves are made up of channels, so these ions can pass through, and it's the fastest way physiologically to create a response. Since we want to create an electrical response, but chemically these ions are chemicals, then the fastest way for us to be able to move these chemicals to create this local electrical potential are through ion channels. But they need certain thresholds before they can activate. They can be chemically gated, mechanically gated, or voltage gated channels. And so graded potentials versus action potentials. So potential will be the potential for an electrical signal to propagate. And graded potentials are different than action potentials in that graded potentials are not an all or none response. So a graded potential might not lead to a response by the neuron. Action potentials actually do. And the reason that is thought why graded potentials don't make a response by the neuron is because of the ion leakage through the cell body as the cell propagates, as the ions through the cell propagate. But once it reaches a threshold level, a threshold electrochemical level within the cell body, then it integrates this response and it sends through the axon hillock the action potential. And so this is the one we're most familiar with, action potentials. This is a response of how an action potential will look. And so it involves these gates of potassium and sodium to take in and out sodium and potassium ions depending on the time. So it also involves the time at which they close. When we look at the resting potential, we can see that this is typically around 61 millivolts, negative 60, or negative 70. And that's attributing to potassium's resting potential. So when we go back to this equation, the reason why we consider all these together and get the resulting value on this graph to be around negative 70 is because the electrochemical potential of the specific ion of potassium has a bigger weight in determining this value, since there's more present within the cell at any given time. And so this is a creation of a graded potential, but once it reaches this threshold here, you're going to have an action potential, which would be an all or none response. And so afterwards, it falls, and it has what is called an absolute refractory period. And so in this region right here, nothing can happen. So the neuron cannot fire again because of this loss of ions. So there's a gradient here. Well, after it passes this absolute refractory period, then you can have a relative refractory period. And so this refractory period could lead to a response. And because of this relative refractory period, if the signal produced during the relative refractory period is actually big enough, then you would have another firing of an action potential. And we can have it in these three regions: resting, rising, falling, refractory period after hyperpolarization, and then finally resting, which is back to the threshold again. Here's an example more in detail of how the different gates or channels are involved when there is a potential. You would have a chemical potential, an electrochemical potential, which would rise above the threshold, reach a peak, and then fall, and have a refractory period. And so within this refractory period, all the channels involved are closed. So there's no transport of ions. Then slowly, the channel starts to open again, and you will reach a relative refractory period. One thing to consider when you're evaluating the different neurons And how action potentials are propagated is that neurons that are bigger tend to propagate faster. And this is due to the less diffusion going along the surface area of the neuron because there's a smaller surface area to volume ratio. Because the thing is, the neuron is simply much bigger. Also, when you have neurons that are myelinated, you're going to have faster action potential propagation. And this is because the myelin sheath is a fatty lipid. So it's going to insulate the neuron, and you're going to have better conduction. Here's an example of how these action potentials propagate through the neuron axon. It's going to propagate throughout the whole axon simultaneously, leaving a zone that's under refractory region, and a zone that's being active, and then a zone that's inactive. And so it would just go straight through until it reaches the synaptic cleft or the synapse. So that would be all for today. Thank you very much for coming.